Chemistry of sex hormones. Estrogen and progesterones. Estrogen and progestins. In normal non-pregnant females, estrogens are secreted in significant quantities only by the ovaries. Although some of it is secreted by the adrenal cortices, during pregnancy, most of the estrogen is secreted by the placenta. There are 3 types of estrogen that are found in significant amounts in the female plasma. This is going to be the beta estradiol, estrone, and estriol. The principal estrogen secreted by the ovaries is beta estradiol. Small amounts of estrone is also secreted, but most of this is formed in the peripheral tissue from androgens converting inside the adrenal cortices. Estriol is a super weak version of estrogen. Its oxidative properties derive mostly from the estradiol and estrone, and this only happens primarily in the liver. Beta estradiol is about 12 times stronger than estrone, and then estrone is stronger than estriol. By far the most important progestin is going to be progesterone. Small amounts of another progestin, which is also known as 17-alpha hydroxyprogesterone, is also secreted along with progesterone and essentially has the same effects. The main function is to get the uterus ready for implantation. Progesterone is also usually secreted in significant amounts during the later half of the ovarian cycle, which is secreted mostly by the corpus luteum. Cholesterol becomes pregnenolone, and then pregnenolone gets converted to 17-alpha hydroxypregnenolone with the help of an enzyme called 17-alpha hydroxylase. The same enzyme also converts progesterone into the 17-alpha hydroxyprogesterone. And then going from both of these 17-alpha versions, it can be converted into DHEA, dehydroepiandrosterone, or it can be converted from progesterone into androstenedione. The conversion between DHEA to androstenedione is going to be with the help of an enzyme called 3-beta-hydroxysteroid dehydrogenase. That will then be converted to testosterone, which then can become any of these other versions of estrogen. The enzyme that converts the testosterone to estrogen is called aromatase. Estrogen and progesterone are transported in the blood, and they're primarily bound to plasma proteins. They bind to albumin with specific estrogen and progesterone binding globulins. They're synthesized predominantly in the ovaries from the cholesterol. Testosterone and androstenedione are synthesized first, and then during the follicular phase, all these androgens and progesterone gets converted into estrogen with the help of that enzyme aromatase. This enzyme is also referred to as CYP19, located inside majority of the granulosa cells. Theca cells, however, lack that aromatase, so they cannot convert androgens to estrogens. They can convert the androgens that will be used to give the granulosa cells to make the estrogen. During the luteal phase, this is where the excess higher amount of progesterone is detected inside the blood. So 2 types of cells. We have the theca cells, which will trigger and respond to LDL receptors, and with the help of stimulation of LH, binding to the LH receptor, that will activate the adenyl cyclase to trigger the second messenger of cyclic AMP. That will then increase the conversion of cholesterol to pregnenolone, pregnenolone to progesterone, and progesterone to the androgens. Don't forget that going from pregnenolone to progesterone, you require the enzyme 3-beta-hydroxysteroid dehydrogenase. And then going from progesterone all the way to the androgens, it has to be converted into the 17-hydroxyprogesterone first, and then into androstenedione before it becomes testosterone. In the granulosa cells, LH also can bind to the receptors, as can LDL, but also the FSH, follicle-stimulating hormone, can also bind. Follicle-stimulating hormone binding to the granulosa cells will trigger the increasing of cyclic AMP, which will in turn activate more aromatase and trigger a lot more conversion of androgens into estrogen. Progesterone created in the granulosa cells can feed back into theca cells and get converted to androgens and then pumped back into the granulosa cells again. So in summary, theca cells have receptors for LDL and LH, whereas granulosa cells have receptors for LDL, LH, and FSH. Here's a quick review of the oogenesis and the ovarian cycle. So the regulation of the ovarian cycle starts off with the hypothalamus secreting GnRH. This will then stimulate the pituitary gland and respond to release more FSH and LH. FSH stands for follicle-stimulating hormone, and LH is luteinizing hormone. Both FSH and LH will then target the ovaries and affect the follicular growth. These follicles will then begin secreting these other hormones. The maturing of the preantral ovarian follicle is going to secrete inhibin in relatively low amounts of estrogen. This creates a negative feedback back onto the hypothalamus and pituitary gland to slow down that tier 1, tier 2 release of hormones. Estrogen plays a role for helping and assisting the development of that ovarian follicle, and now as that follicle grows, it is dependent upon the FSH and LH to mature and then rupture. As a mature follicle produces large amounts of estrogen, this stimulates a positive feedback, and that then triggers the LH surge. Without this surge, without the LH, there cannot be any ovulation. After ovulation occurs, and if there is no fertilization, the corpus luteum will then form over the ovulated follicle. The corpus luteum secretes large amounts of progesterone, estrogen, and inhibin, and it inhibits the hypothalamus and pituitary and rebuilds the uterine lining. Over the next 10 to 13 days, if not fertilized, begins to degenerate, and the cycle will then repeat itself where the LH dips back down by negative feedback, and levels of estrogen, progesterone, and inhibin also begin to drop down. Inhibin is the hormone that reduces the amount of FSH. The green arrows represents a stimulation, so that means it increases the amount of that target cell. So GnRH targets the pituitary gland in the anterior side to release more FSH and LH. That will then cause the maturation of the preantral ovarian follicle to secrete inhibin. Inhibin will then inhibit the production of FSH. Estrogen levels are quite low at this point. As that antral follicle develops, now it is dependent on how much FSH and LH is secreted. So as a mature follicle produces larger amounts of estrogen, this will then stimulate the hypothalamus in a positive feedback manner to release more LH. The LH surge from the pituitary gland induces ovulation, rupturing of the ovum, and the releasing of the egg. The corpus luteum is then formed under the influence of the LH, and if there is no fertilization, and then the corpus luteum secretes large amounts of progesterone, estrogen, and inhibin, which will then inhibit the hypothalamus and pituitary gland. Because there's so much of that, then it's going to slow down the need for any more LH. And then over time, if there's no fertilization, it regresses into the corpus luteum and then the corpus albicans. Here are the key functions of estrogen. It affects on the female sex organs, increases the sizes of the ovaries, the fallopian tube, the uterus, the vagina, the external genitalia. It can alter the vaginal epithelium, transitioning it from a cuboidal to a stratified type. It also has effects on the fallopian tube by increasing the number of ciliation, so that increases the movement of the egg. It also has effects on the breast tissue by developing the stromal tissue and ductile system. One note to be aware of is that progesterone and prolactin are required to cause the functions of lactation and the full development of these structures. Estrogen also plays a role on the skeletal system. It inhibits the osteoclastic activity in the bones, therefore it actually boosts growth. That means during menopause, when the estrogen dips, the bone breakdown occurs at a higher rate. Effects of estrogen on the skin. Estrogen also causes skin to develop texture that is a little bit more soft and smooth. It increases body metabolism, but it does increase fat deposition, and this is also why females tend to store fats in certain areas of their body over male counterparts. Functions of progesterone. Progesterone has anti-estrogenic effects on the myometrial cell. Its main function is to decrease the excitability of a hormone called oxytocin, which triggers contraction of the uterus. It also in part reduces the number of the estrogen receptors and the LH secretions. One key function is that it increases the conversion of the 17-beta estradiol into the weaker counterparts of the estrogen, estrione and estriol. The main function is to prepare the uterus for implantation for the fertilized egg, and it makes sense because it does not want to lose the fertilized egg. Progesterone promotes the changes inside the uterus and fallopian tubes, and is predominantly secreted by the corpus luteum, placenta, and small amounts of it in the follicle. It promotes changes in the uterine lining because the whole purpose of this hormone is to prepare the uterus for implantation of the fertilized egg, and this happens in the later parts of the cycle. It decreases the frequency and intensity of the uterine contraction so that the implanted egg is not going to be exposed out. Progesterone promotes the development of the lobules in the alveoli of the breast and causes the alveoli cells to begin proliferating, enlarging, and becomes more secretory. However, it does not directly cause the alveoli to secrete milk and lactation. The milk secretion is only prepared after further stimulation via sound and suckling of the nipple and also in combination with prolactin from the anterior pituitary gland.